All right. Hello, and thank you for joining us at MIDDD Plus 2023. In this session, Deanna Mudie, Principal Scientist of Research and Development from Lonza, will present on using in silico PK simulations for early formulation development of amorphous solid dispersions. As a reminder, you may ask questions via the Q&A tab on your dashboard at any time. If you need any technical assistance at all, please use the chat tab. Before I pass things off to our speaker, we have one quick poll question for you today. Let's see if I can get it to go. I cannot. I cannot. So Deanna, take it away. Appreciate it.
Great, thank you so much, and thanks for everyone for attending today. So I'll start my talk going over some key design considerations on amorphous solid dispersions, which I'll be abbreviating as ASDs throughout the talk. I'll then go into Lonza's approach for in vitro and in silico bioperformance assessment of ASD formulations. However, I will be spending most of my time on a case study of how acalabrutinib ASD tablets were overcoming a pH effect in vivo. And then I'll summarize, highlighting how in silico PK simulations can be used to de-risk ASD formulation development. So ASDs can enhance the bioavailability of poorly soluble drugs. And poorly soluble drugs, as most of you know, make up the BCS2 and 4 categories of the biopharmaceutics classification system. And these compounds are still highly prevalent in drug development pipelines, making ASDs really important. So ASDs work because the amorphous form of the drug has a higher apparent solubility than the lower energy polymorphs or crystalline forms. And this amorphous form can therefore boost factors such as dissolution rate and really allow for supersaturation in the gastrointestinal tract, which helps to drive that rapid absorption. However, the amorphous form is thermodynamically unstable, so we must formulate it as an ASD with a concentration sustaining polymer in order to stabilize the amorphous state, both in the solid state and in solution. And so when we're designing these ASD formulations, we really need to keep all of the key attributes in mind. So it's not just that stability, but also other factors such as in vivo performance, manufacturability, and drug loading. And to do that, we need to not only think about the ASD intermediate, but also the dosage form. So we put the ASD in common dosage forms such as tablets, capsules, and suspensions. And some of those key formulation factors that we have in mind include the drug and the excipient phys-chem properties, the drug and excipient loadings, and also factors such as ASD particle properties. These ASD formulation factors, as well as the physiological factors that really drive bioperformance. So when an amorphous solid dispersion is ingested into the stomach, when it's designed as an immediate release formulation, it is designed to disintegrate and dissolve. And it dissolves into many different drug species depending on the API and formulation properties. So we can often get these drug polymer colloids, which can be important for absorption, as well as drug associated with the endogenous bile salt micelles in the GI tract, as well as amorphous drug in the free unbound form. And it's these different species that can contribute to absorption across the intestinal membrane, which is really what we're striving for when we're developing these formulations. And in really simple terms, what we're looking for is drug concentrations that are reached very quickly. So we're looking for rapid dissolution, but we're also looking for good sustainment. So we really want to maximize these supersaturated drug concentrations such that we have this immense solubility boost over the crystalline form shown in green here. Things that we look out for in terms of formulation is to avoid inadequate sustainment. So we want to avoid rapid precipitation to the crystalline form. And we also typically want to avoid inadequate dissolution rate, so slow dissolution to lower sustained drug concentrations. So when we're designing these dosage forms, it's highly important to have methods to, of course, determine whether or not they're working, whether or not we expect them to perform in vivo. And traditional methods alone are typically not predictive of ASD bioperformance. So compendial dissolution methods such as USP1 and 2 methods are highly useful in some areas, particularly for quality control dissolution testing. However, these don't simulate the physiological environment, which is very complex, often has much lower volumes, much more complex buffer species and components, as well as different absorption components. So it's really important that we don't just rely on these single phase tests, but we use more complex in vitro tests in conjunction with in silico models, as I'll go into next. What we do to circumvent this issue is to use an in vitro performance toolkit for assessing ASD bioperformance. So we have a number of tools. These are just a few of them that are designed in some cases for early ASD formulation development. So these are really high throughput material sparing screening tests that are used to develop the ASD intermediate in the first place, determine parameters such as amorphous solubility and choosing the best concentration sustaining polymer, for example. Moving along into actually understanding the dissolution performance of those ASD intermediates. And then finally looking at some lower throughput, but still tests that are designed for the ASD dosage form, such as a tablet or capsule. So we still do use USP2 apparatuses quite a bit, and sometimes just in a transfer test mode, as I'll go into in a later slide, as well as more physiologically based dynamic tests, such as the controlled transfer dissolution test, which I will also talk about. And as we use this toolkit, we're always choosing our test and our parameters based on the in vivo problem statement, so our formulation and the targeted physiology of interest. But what we find the most powerful is when we take these in vitro inputs and we combine them with in silico tools in order to really de-risk our program and make good formulation development decisions. So we take inputs such as calculated properties from GastroPlus ADMET predictor, which other speakers have already talked about today. They've talked about the utility of ADMET predictor. So we take these calculated properties as well as measured in vitro drug properties together with these in vitro dissolution data in order to make these. In some cases, in later stages, we do also have in vivo PK data that we can incorporate, but we still find it powerful to generate these predictions even in the absence of the in vivo PK data. What we're getting from this are things like parameter sensitivity analyses to help us really understand key formulation and physiological factors that could be high risk for our compound. We use these to identify physiological barriers to absorption, so really define the problem statement. We also use them for formulation and in vivo study de-risking, which I'll talk about in the case study, as well as guidance for any additional testing that we think is important. And so with that, I'll give a case study really showing how we put the in vitro in silico development process to use. So the marketed drug Calquence, at least as it was originally developed, was developed as a crystalline acalabrutinib in a capsule. What happened with this product was that it showed reduced performance in the presence of acid-reducing agents. This is a BTK inhibitor indicated for oncology. So quite a large fraction of oncology patients are on these acid-reducing agents, which makes this DDI problematic for them. So based on the FDA label, the plasma AUC of acalabrutinib is reduced by 43% when taken with a proton pump inhibitor. And so therefore, patients must avoid taking these PPIs or other acid-reducing agents, which I abbreviate as ARAs, when taking acalabrutinib. So it really acts to undermine efficacy of the drug for these patients. And the reason that this happens is because acalabrutinib is a weak-base drug. So it has high solubility at gastric pH under normal fasting conditions. But then when patients are taking ARAs, their gastric pH is elevated. And it's in this region that crystalline calibrutinib has much reduced solubility. And this reduced solubility leads to the decreased bioavailability in the presence of ARAs. So we decided to create an ASD tablet to overcome this pH effect. So is a biopharmaceutics classification system 2 compound, again with a low solubility at these elevated pH values and a relatively high permeation rate. This is actually the solubility pH profile that was measured for crystalline acalabrutinib by Pepin et al., published in the literature, and then for an amorphous solid dispersion intermediate that we measured in-house. And as you can see, there's about a 7-fold solubility enhancement of the ASD over the crystalline form at elevated gastric pH. So this looked to be a promising candidate for this drug. And in terms of its candidacy for, being a good amorphous solid dispersion formulation, based on our technology map here that looks at crystallization tendency of the API versus its lipophilicity, it fits into this high drug loading region. So this is a region where we would expect to be able to achieve a relatively high drug loading and have relatively low risk in terms of crystallization tendency when making an ASD. So the way we designed the tablet was we ended up, after some formulation development activities, choosing a 50% active loading with the dispersion polymer HPMCASH in an ASD, and then we put that ASD into an immediate release tablet. This isn't the focus of my talk today, but we did a physical and chemical stability analysis of the ASD and tablet and found it to have good physical and chemical stability. And due to that high drug loading that we were able to achieve, it was actually 60% smaller than Calquence capsules, which was beneficial from a patient compliance perspective. In terms of our in vivo study goals, we decided to use a beagle dog model to study the ASD versus the Calquence capsule. Our goals were to mitigate the pH effect using the ASD tablet. Our goal was also to match plasma exposure of fasted Calquence when using this ASD tablet, and we wanted to show the pH effect with Calquence as a control in our study. And so as I mentioned, we used an in vitro in silico ASD development strategy for this program. We first did a polymer screen where even before making an ASD intermediate, we were able to choose the best concentration sustaining polymer, which turned out to be HPMCASH. So we moved that forward as one of our formulations for which we manufactured ASDs. So we manufactured a number of ASD formulations, in this case by spray drying. And then we tested small-scale ASD dissolution in order to select our lead ASD intermediate. Once we did that, we made immediate release tablets and we tested their dissolution. We then did our in silico predictions using GastroPlus, which we used to de-risk and to design our in vivo study in beagle dogs. And today I'm going to be focusing on this in vitro tablet dissolution, as well as the in silico predictions and the in vivo study results. So with our ASD tablets was that they were able to achieve our performance goals in vitro. So I'm showing an example here of our controlled transfer dissolution test. This is a very similar test to other tests in the literature, such as the artificial stomach duodenum or the gastrointestinal simulator. This test is designed to capture dynamic movement of the dosage form from a simulated gastric to a simulated intestinal compartment using relevant fluid compositions and also gastric emptying rates. So in this test, either the ASD tablet or the Calquence capsule in either a pH2 HCl medium designed to reflect fasted dogs, or in an elevated pH 6 gastric medium designed to reflect dogs on an acid-reducing agent. We then were able to use the system to pump dissolved drug and drug solids into the intestinal compartment, which contained a pH 6.5 medium with simulated intestinal fluid, so these bile salt micelles, and we were able to monitor concentration versus time in these two compartments. So results of these tests are shown here. For the gastric pH2 test on the top, this is the concentration versus time profile in the gastric compartment for the ASD tablet in blue and for the Calquence capsule in green. And then this is the concentration versus time profile in that simulated intestinal compartment, again, for the two dosage forms. So as you can see, the ASD tablet was able to match performance of the Calquence capsule as desired, and both dosage forms supersaturated in the intestinal compartment, achieving relatively high concentrations. And this was expected because the crystalline Calquence has a very high solubility at pH 2, and therefore we achieved similar results between two dosage forms. However, the story is a little bit different when we get to the pH 6 gastric media. So in this case, you can see that the ASD tablet dissolves to a much higher value than the Calquence capsule. That's due to the much higher solubility of the amorphous form compared to the crystalline form at high pH. And then upon transfer to the intestinal compartment, again, we see the ASD tablet is able to supersaturate, whereas the Calquence capsule dissolves to its crystalline solubility in the test. And so as you can see, the ASD tablet achieves similar concentrations at pH 6 as it does to pH 2, which would suggest that it has the potential to mitigate the pH effect. And I should also mention, even though you're seeing these decreasing concentrations versus time, it's not due to precipitation, so we did not see any precipitation in the test, but it's due rather to the fact that we're pumping the drug and dissolved particles into the next compartment. So we were really happy with those in vitro results, but we wanted to use GastroPlus in order to gain much higher confidence going into our in vivo study. So we had set up an in vivo study in beagle dogs, and we wanted to go in with the best formulation with the best dose that we thought would really help us achieve our study goals, of course, without having to resort to reformulation or doing multiple studies. So we use GastroPlus 9.8, and we use the default fasted beagle dog physiology in GastroPlus. However, we did make one adjustment. This was to adjust the gastric pH to reflect either a pH 2, so that would be a fasted treatment. To achieve this in the in vivo study, we actually gave the dogs penagastrin in order to simulate a low pH, or we adjusted it in the other simulation representing an ARA to a gastric pH of 6. And this was to reflect the famotidine pretreatment in the dogs designed to raise their gastric pH. And then as I'll go into in the next slide, we further adjusted this gastric pH to account for surface pH effects, which can have an important effect on dissolution rate. We were able to take PK parameters derived from the literature from a published oral solution formulation in dogs, which use single compartment PK. So we use that as a basis for our clearance and elimination factors in the model. We used physicochemical properties that were measured or calculated, again, from that Pepin et al. Source, FDA documentation, and ADMET predictor. We did a measurement of solubility versus pH profiles in-house for the ASD, or again, measured values from Pepin et al. And the literature were taken for the simulations. Also, as I'll go through in a coming slide, we were able to take in vitro dissolution testing using a USP2 transfer test to determine or to really translate those in vitro dissolution rates into in vivo dissolution rates for our simulations. And then we assume negligible precipitation based on our experience with our in vitro testing as well as published literature data. And I really wanted to highlight that these were bottom-up predictions, so we did not do any optimization or any curve fitting. They were really used as a priori predictions going into our in vivo study. So now spending a little bit of time on this in silico input of adjusting gastric pH for surface pH effects. So when acalabrutinib dissolves, it can tend to raise the pH of the solution. So this is a plot of the saturated solution pH of acalabrutinib versus the starting bulk pH. So you can see if we start with a bulk pH of 2 using hydrochloric acid, once the acalabrutinib dissolves and achieves saturation, the drug actually raises the pH to about 4 for the crystalline drug and to about 4.6 for the amorphous drug. And this is really important because this saturated solution pH can act as a surrogate for the estimation of the pH at the surface of a dissolving drug particle. So at the surface of a dissolving drug particle, we expect the fluid to be saturated. And it's this saturated solution pH or the surface pH that is really driving the solubility of the drug from the surface and really dictating that dissolution driving force. So it was really important to us to reflect an accurate solubility or solid surface concentration in the simulations. So in order to achieve that, we took our simulated bulk pH, so either a pH 2 for fasted conditions or pH 6 under an ARA, and then we adjusted that to the measured surface pH for the ASD tablet, which we measured in-house, or from the published values by Pepin et al. And again, what this allowed us to do was to get a better, more accurate prediction of what we thought the dissolution rate would be. Next, we needed to account for in vitro dissolution rate itself in the model. We did this using the Z-factor equation and GastroPlus, and we fit the Z-factor equation to measured in vitro data from a USP2 transfer test. So in this test, we placed either the ASD tablet or the Calquence capsule in a gastric medium, so at either pH 2 or pH 6 hydrochloric acid. Then at 30 minutes, we added a concentrated intestinal solution to simulate an intestinal medium. We monitored concentration versus time during this test and then plotted it here. So this plot has the pH 2 gastric results, and this plot has the pH 6 gastric results. So as you can see, we got, again, rapid dissolution of both dosage forms at pH 6, followed by sustainment of these supersaturated concentrations. So what we were able to do was to fit the Z-factor equation to this profile here to extract the Z value for the pH 2 gastric dissolution. And then we did the same thing at pH 6. So as expected, we see the slower dissolution to a lower concentration under pH 6 conditions. We were able to extract Z factors here, as well as Z factors for dissolution in the intestinal medium, which was pH 6, containing bile salts and lipids. And what extracting the Z factor allows us to do is really translate these in vitro dissolution rates to a simulated in vivo rate using a lump term which takes into account factors such as effective diffusion coefficient and particle size. So what we found was that our in silico predictions suggested that the ASD tablets indeed would achieve our study goals, which was to overcome the pH effect. So you can see in these simulated profiles that the ASD tablet achieved similar profiles at both low and high gastric pH. So AUCs were within 2%. We were able to match exposure of the Calquence capsule. So the ASDs here for Calquence capsule at low pH compared to the ASD tablet were within 10%. And then we did indeed expect to see this differentiation between low and high pH for the Calquence capsule. So the Calquence capsule AUC was about 3-fold lower under these high pH conditions. And our simulations were very accurate, and our in vivo outcome was actually matching very well the in silico prediction. So as I mentioned, this was a beagle dog study. This was at a 100 milligram dose, where we treated the dogs with either pentagastrin to achieve low gastric pH or famotidine to achieve high gastric pH. So the smooth lines here are those in silico predictions I showed on the last slide. and then the data point showed the actual values. So you can see that the simulations match the actual values very well. And when you look at the absolute average fold error, which is a way to measure how closely the simulations followed the experimental data, we got values less than 2. So a value of 1.0 would mean we matched everything 1-to-1. And the fact that we're seeing less than 2-fold for AUC is very, very promising, especially for bottom-up predictions where we did not do any optimization. CP time, so the concentration time profiles themselves, matched very closely for these simulated profiles for ASD tablet and Calquence at low gastric pH. And then we didn't do quite as well for the Calquence capsule at high pH, where there was still a little bit of an over prediction, but still we were within what we wanted to achieve. And so with that, I would like to summarize. So first and foremost, the ASD tablets were able to overcome the pH effect on the first try using this in vitro in silico development approach. Our in silico PK simulations successfully predicted our in vivo study results using a bottom-up approach. And our in silico PK simulations, we think, can really be used to accelerate ASD formulation optimization and reduce costs. So as I mentioned before, we can design and simulate our in vivo studies before we perform them to go in there with our best foot forward. And we can also use it for things like parameter sensitivity analysis and in vitro test design. I also have a few references here. So we published our results online, and you can use this QR code to access those publications. And finally, I would like to acknowledge many of my colleagues, in particular Aaron Stewart, who performed the bulk of those simulations, as well as many other colleagues who were instrumental in performing the in vitro testing and performing the study design and interpretation. So,
Deanna, thank you so much for a wonderful and informative presentation. Thank you all for your participation today and for the great questions and Q&A. Unfortunately, due to the slightly late start, which was on my end, we'll have to shelf your Q&A questions for the Q&A session at the end of this track today. So please bring those questions to the track and we will address them later today. Thank you so much, Deanna. Much appreciated.
thank you.
I hope the rest of you join our next presentation. Thank you so much.